Yeah, however possible.
Can we park at the car place? That's like the beginning of the street.
So you guys are now live on YouTube, so just, you know, mind what you say. Oops, sorry.
I'm gonna stand, I think.
You're just like filming me on YouTube.
So, no, now I'm feeling.
YouTube now.
All right, you guys are YouTube live. There you go.
Nabil PhD Defense.
Your mom says, God bless Nabil and his ants and leafhoppers.
Tree hoppers, but you know.
Yeah.
All right, here you go.
I'm gonna stand.
Yeah.
I love it.
Front row, Carl. Ex-roommate VIP treatment.
I'm like, good. It's faded a little bit.
Brian, there's a seat up front if you'd like.
Well, since we can't fit any more people in, we should stand here. It's really an honor and really a pleasure to welcome you today to the Nabil Nasseri's PhD Defense. And Nabil came to us from doing a master's at Georgia Southern University, where he worked with Bruce Schulte on elephant chemosensory behavior and the effects of elephants in modifying tree architecture on the herpetofauna of mostly acacias in Tanzania. And then after that, he went to Drexel University. He took a job with Drexel University, a study abroad program, and worked in Equatorial Guinea. And just as he finished that program is when I first met Nabil in 2011. He texted or cell phones, he didn't text or cell phone me, most people know that. My then master's Joe and I are having a little thing here about cell phones. For those of you who aren't in the know, I'm a rabid anti-cell phone person. Anyway, Nabil got in touch with Renee Petipa, who was then a master's student with me, and she and I were in the field at Impala Research Center in Kenya. And lo and behold, he was visiting Nanyuki, the stinky little town at the base of Mount Kenya, and could he join us in the field for a week or two or something like that. And it was a real pleasure to be in the field with him. And Nabil came here intending to continue the work and really branch it out, the work that we had started in Kenya, looking at the effects of a large ecosystem engineer, East African elephants, and termites on the invertebrate communities. And he came back here and joined us that fall and wrote many, many, many grants. And as we all know who've tried that, it's very difficult to get funding, and he very wisely switched his program from looking at charismatic megafauna to charismatic microfauna, and thought about how ecosystem engineers affect the communities in which they're embedded here locally in the United States and in Texas on mesquite trees, which he's going to tell you about today. So I'm not going to preview that for you, but just before we go there, I want to say that Nabil is an extraordinary photographer, and I'm sure many of the graduate students in this room will be glad when he is no longer submitting his photographs for our yearly competition. And he's also quite an extraordinary cartoonist. He loves to put his ideas down in these flowcharts. And this is my favorite cartoon of Nabil's, depicting our lab. And that's me up in the left-hand corner with a coffee cup saying, "What's your hook?" And some of you might recognize this creature in the middle. my very nervous dog. But I also want to say that Nabil is, without a doubt, the kindest, most generous person I can say I have ever met. Nabil will always be there for you to do whatever you need. If you need help collecting salamanders, or you need help collecting lizards, or you need help collecting worms, or you need help collecting small mammal trapping.
Moving every
Moving
refrigerator in MLS.
Nabil will step up to the plate, and it's truly been a pleasure and an extreme honor to have him as my PhD student. And so, without further ado.
Now to live up to the hype. So thank you everyone for coming. This is basically the culmination of about 6-7 years of work, which allowed me to really grow as a scientist. I remember when I applied for Allison's lab, you know, I asked her what kind of graduate students she looks for or is looking for, and she's like, her main kind of stipulation is you're not allowed to work in her system or under her questions, because she believes the whole point of a PhD is for yourself to grow as a scientist. And at that time, I didn't quite understand what that meant, but I still was like, you know, I'm excited with my project and I'm sure I can figure it out. And I think that type of mentorship has truly helped me. For my master's, I got in and I didn't even know what a grant was. I figured I'd take loans out to pay for my research. I was very uneducated in graduate school through, you know, the lack of training I got, or in my undergrad, or just lack of experience. And then I came here thinking, oh, I was on NSF for my master's, money's easy to get. Not true. But this project, another thing Allison taught me when I first came here was, as a scientist, there's two ways to do science. There's one where you just love an organism or a place, and you could care less what the question is, as long as you get to work with that organism. Or you can, or another type of scientist is you really care about particular questions and you don't care what the model organism is. When I arrived, I was very kind of tunnel vision, elephants, 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 that's all I want to work with. However, not getting funded was kind of a blessing in disguise, because it allowed me to fully step out of this kind of realm of focusing on a single organism, and then broadening my scale of thinking onto whole ecosystems and community level interactions, which is kind of what my passion is now, although I still would like to incorporate elephants into that. But I remember I was doing some transect work during my master's, bunch of acacia trees cutting me, trying to find acacia trees. And I made a promise to myself there and then, after like mile 10, I was like, I am never working with trees. And then the most painful injuries I've encountered because of wildlife have always been insects. I got knocked down to my feet by a walking stick bug squirting acid in my eye. I got stung by a caterpillar in Equatorial Guinea and thought I had been bit by a bush viper. It basically gave me enlarged lymph nodes, high heart rate. I was like, no insects. So lo and behold, I am going to be talking to you about my PhD on the direct and indirect effects of anti-hemiptera mutualisms on the invertebrate community and their host plant, honey mesquite. So trees and insects. I have tons of people to thank for basically getting me to where I am now, especially my committee members, Sarah, Nick, Joseph, and Allison as my advisor have really helped me with everything. I had tons of help in the field from just friends and family coming out there whenever they had time. I have Sarah here first because pound for pound she was arguably the best person to have in the field with me, because when I'm in the field I go 100 miles and I don't stop. And she had the same mentality. Was when we did field work, we were working hard. but also we had a lot of really good, you know, just jokes and laughs and camaraderie. And she's about 5'2", but would carry, you know, 4 T-posts and a bale of, you know, barbed wire walking through the field with me. So really a lot of thanks for her for the field work. I had tons of undergrads working for me in the lab with insect identification and counting seed pods. And a lot of them came and went. Others stayed for a couple of years. Luke McMartin, Luke was actually stayed for 2 years and got an Apple grant to do a small satellite study here in Vermont on anhemipteran mutualisms on goldenrod, and is in the process of trying to get that published now. All my funding sources, so from small organizations to businesses to scientific societies, UVM, and then especially Welder Wildlife Foundation. So this is where I did my research. They provided a fellowship for me to support me during the summer, as well as providing some funding for field work. So just a quick overview of what my project entailed, or kind of the landscape of this talk is I'll first focus on the importance of mutualistic interactions within communities, then moving on to community level effects of mutualism. So how to test community level effects, looking at direct effects and indirect effects. And the objective of my PhD was to determine the effect of anhemipteran mutualism on invertebrates, so looking at the community composition at the family level, as well as the community composition on functional guilds. And I will explain or define more concisely what I mean by community and what I mean by functional guilds. So as ecologists, we are fundamentally interested in understanding the relationships between organisms and the impacts they have on one another, and then as well as how they may impact or alter their surrounding environment. And our primary focus has generally been on how species interactions structure the overall community composition. So how does species A and B interact, and then how does that impact each other, or how does that impact a particular resource? So so here we're looking, so community I've defined as a group of interacting species occurring together in space and time. So for the sake of my study, my interest is looking at the effect of the anhemipteran mutualisms on a community structure, so would it make sense for me to look at insects that never interacted with hemipterans per se. So in this case, whenever I mention community, these are groups of interacting species that are occurring in the same space. So as ecologists, we're primarily focusing on direct antagonistic interactions that result in one species benefiting and another species suffering. So particularly what we focus on are predation events or competition as our major drivers in structuring communities. And we focus on these interactions because they result in a very concrete ending. You have two organisms, they either compete or one eats the other and you lose that species. So it's a very definitive interaction that we see. So one's consumed or one's displaced. However, mutualistic interactions, and these are interactions in which both organisms gain a net benefit from interacting with each other, and they provide some sort of service for a benefit. And mutualisms are pretty ubiquitous in most systems and can alter predation or competition outcomes. So for example, birds form a facultative or non-obligate mutualism with some mammals, and it allows them to be more effective predators. So here we have a cattle egret sitting on a horse named Tadpole. And this is a form of mutualism in the sense that the cattle egret is utilizing this horse as a food patch. So the cattle egret can hang out on the horse and pick off insects that are coming to feed on the horse. And in return, the horse receives a benefit in the sense of it doesn't have biting insects all over it. And this is found not just, on one horse in Texas. We can see this all the way in Africa. So here you have red-billed and yellow-billed oxpeckers that have formed this mutualism with a Cape buffalo. And what they do is they just hang out in the Cape buffalo, feeding on ticks and things like that that would otherwise be parasitizing the buffalo. And then the buffalo gets that additional benefit of getting groomed. And these are found not just terrestrially, they're found in aquatic systems. So different organisms, same mechanism. We see this with cleaning shrimp and certain fishes and corals. There's even a mutualism between crocodile opens its mouth out, a bird comes in and picks out all the scraps of meat out of its teeth. And you have basically a bird dentist, and the crocodile in turn doesn't eat the bird. So you have these mutualisms that span multiple organisms and multiple habitats. And in addition, mutualisms can potentially dampen or enhance these strong community structuring interactions like predation or competition. For example, here we have Acacia drepanalobium and Kermanagaster ant. So this is our classic ant-acacia mutualism. And what we have here is this is a tightly co-evolved mutualism. So unlike the cattle egrets and the oxen or horse, these are obligate to one another. So these acacias are one of the favorite foods of mega herbivores in certain savanna ecosystems, particularly elephants and giraffes. And if you to remove the ants, these things get decimated and they can't really survive. However, through employing these ants, they receive a really strong protection benefit, so therefore these ants are able to defend them from elephants or giraffes feeding on them. And the way they keep these ants around, or they form this really tight mutualism, is the acacias produce what we call extrafloral nectaries, which are glands that open up, or they're bowl-shaped along the petioles of the leaves, and they fill these up with carbohydrate liquid. So nectar, but specifically it's not for pollinators. The sole purpose is for attracting ants. And then they also basically sweeten the deal by providing domatia. So domatia are these modified thorns that they hollow out, and then the ants use those as nesting sites. And in return, the ants provide these protection benefits because they want to protect their home and they want to protect their food source. So these are highly co-evolved, and we see this in Africa, but we can also see these with bullthorn acacias in South America. So again, these mutualisms showing really strong interactions that have some form of selection on them. And they can also, from a sense of competition, when Acacia drepanalobium, at least in the savannas in Kenya, these are monospecific areas. So Acacia drepanalobium is the only standing tree. And it's potentially because they've employed these ants as a defense, so therefore all the other trees that have tried to recruit there get hammered by all the other herbivores because they're not protected through this mutualism. So as our understanding of the effect of mutualisms, or as our understanding of the effect mutualisms have in their community grows, so does the perspective of how these mutualisms can alter their communities. So for my first objective, I want to determine community level effects of mutualisms. So first, looking at ant hemiptera mutualisms as a model system. So ant hemiptera mutualisms are a good model system, as ant-based mutualisms as ants are typically really strong interactors or key players in the communities that they're found in. So ants are are capable of altering soil properties through massive nest building. So this is leafcutter ants at my field site in Texas, and there's Emily checking them out. They are important seed dispersers. So this is a red harvester ant collecting seeds. This is not to scale. And then they're also often key predators. And the key predators, so here we can see a Camponotus running off with a inchworm. However, through engaging in mutualisms, these kind of key characteristics can be enhanced or amplified, and therefore having a stronger effect on their communities. So ant-hemiptera mutualisms are food for protection mutualisms. So these are ants receive a carbohydrate reward. So just like in the acacia ant mutualism, same mechanism, the tree hoppers provide a liquid carbohydrate called honeydew. So they excrete this liquid and in exchange for protective services. So here we can see an ant, Camponotus, tending tree hoppers. So this is an adult, and this is a nymph. And you can see here this honeydew being excreted. So these tree hoppers are phloem feeders, so they're basically taking up phloem sap from the plants, and therefore since the primary of their food intake is carbohydrate rich, they excrete carbohydrate rich excrement, which the ants collect. And therefore, again, this is in exchange for protection from predators, parasitoids, and even potential herbivore, competing herbivores. So these herbivores might either be other hemipterans that are looking for a nice spot to feed, or they could be other herbivores that are competing for nest sites or just areas to hang out. So this was, I was lucky to grab this picture because it kind of solidifies this mutualism in a nice picture. So here you have an ant tending a nymph as this ant is carrying away that same inchworm. And a lot of studies have shown that when ants are engaged in these mutualisms, their activity increases as well as their aggressiveness. So they become pretty indiscriminate in their aggressive behavior. So without the tending ants, they may encounter another insect and leave it be, but if they are tending and they encounter another insect, they're usually attacking that insect to try and catch it or to knock it off. And for the rest of this talk, whenever you see a blue arrow will indicate a positive interaction, a red arrow will indicate a negative interaction, and if they're solid, these are direct interactions, so species A interacting with species B. If they're dashed, it's an indirect interaction, so A impacts C through altering species B. And these mutualisms, just like the cattle and bird mutualism, are pretty ubiquitous. They're found everywhere. So I'm just going to take you on a road trip from the East Coast to the West Coast. So here are ants tending hemipterans on goldenrod on the side of a road in Richmond, here in Vermont. Here are woolly aphids at a campsite in upstate New York. Here are mealybugs on mimosa that I found at a mimosa tree in the parking lot of a Cracker Barrel in Mississippi. Here we have, these are fire ants tending a scale insect. And this was, I was out riding my bike with my friends through my old neighborhood, and there was a bunch of grass, like lawn clippings. And I was just starting my PhD, and I was like, hey, let's check out these clippings. And sure enough, I looked under this, cut down oak tree branch and found the mutualism there. And then here I was taking photos from my friend's wedding, and I was like, hey, hold on, guys. We were in a walking trail because I saw some ants, and I followed them, and sure enough, I failed here as a field naturalist because I didn't have my field lens. I couldn't tell exactly what they were tending, but I think they may have been a type of thrip. But here, all the way in California, we can see the mutualism occurring there as well. So, and it's not just in the States. These are basically anywhere ants are found, this mutualism occurs. It's from papers I've read, anywhere from 41% to every ant will engage in this mutualism if it's available to them. So this, due to its kind of how common it is and the potential impact they can have, they've been studied pretty highly. However, most of the studies just focus on their interactions between the partners. So what benefits or services do they provide? But we still lack an understanding of their community level effects. So therefore, the focus of my PhD has been to simultaneously measure their community level impacts by sampling their effect on the arthropod or the invertebrate community, as well as looking at the subsequent effect on their host tree. So these mutualisms always have a host tree that they live on, and presumably all of these interactions should be, so we're going to say all these interactions should interact, but should be feedback into each other. So my first kind of overarching objective was to determine the direct effects of the mutualism on the community. So specifically, what effect of ant hemiptera mutualisms have on the invertebrate community, and I'm going to be measuring this or testing this at the family level as well as on functional guilds. So functional guilds are a way to bin your species based off of organisms or species that share a common resource. So in my case, these are functional guilds are going to be based off of what they feed on or what their food sources are. So tending ants should have this negative association with their arthropod community based off of the aggressive tending behavior or protective behavior of the ants. So if the hemipteran's main benefit comes from the ants' indiscriminate removal of other invertebrates, then we should expect lower abundances of invertebrates on trees that have the mutualism versus trees that don't have the mutualism. And then this leads to my second objective. And again, just to recap, red arrows, negative interactions. So for my second objective, I'm interested in looking at what are the indirect effects of the mutualism on the community. So this is going to be looking at the effect of ant hemiptera mutualisms on their host plant. And I'm going to be measuring reproductive success and using flower and fruit counts as a proxy, and then quality using condensed tannins and percent nitrogen as proxies. And therefore, if we're expecting the mutualism to have this negative interaction with the arthropod community, then we should also expect them to have a positive indirect impact on their host plant, because through their tending behavior, not only are they removing predators, parasitoids, but they're also removing herbivores. So through the reduction of these herbivores, we expect to find these trees to be higher quality and have higher reproductive success. And these will be an indirect interaction because this tree is benefiting through the ants removing harm or much more damaging herbivores. So therefore, in the presence of the mutualism, host plants should be higher quality and have higher reproductive success. So just to kind of recap everything, my first objective is what are the direct effects of ant hemiptera mutualisms on communities? So the null expectation would be that invertebrate communities are not associated with the mutualism, meaning that whether the mutualism is there or not, you'll find the same community, same abundances. We hypothesize, however, that the invertebrate community is negatively associated with ant hemiptera mutualism. So therefore, in the presence of ant hemiptera mutualisms, we'll find lower abundances of invertebrates, then an N-hemipterin present, then if N-hemipterins were not present. And then again, we measured the invertebrate community and looked at it at the family level and on functional guilds. And my second objective was what are the indirect effects of these mutualisms. So the null would be hosting N-hemipterin mutualisms does not affect the host plant quality or reproductive success, whereas we predict or we hypothesize that mutualisms do affect the host plant quality and reproductive success. So therefore, in the presence, we'll see higher quality and higher reproductive success. And again, we measured this looking at condensed tannins and percent nitrogen for quality, and then flower and fruit production as proxies for reproductive success. So this work was done at the Robin Bessie Welder Wildlife Refuge Foundation. It's roughly 3,200 hectares, and it's managed for rangeland and wildlife experiments. Luckily for me, when I was there, it was me and pretty much one other grad student doing research out there, so it was kind of open terrain. And just to give you an idea of what the landscape looks like, it's characterized as a mesquite grassland. So this is grassland, scrub savanna, pretty dry. And you can see here a lot of basically open grass with clumps of trees. And the dominant tree species there is honey mesquite. So, and then kind of the players or the mutualism that I'm interested in are Campanota sayi and Vendusiae segmentata. So Vendusiae segmentata is a membracid, and common names are tree hoppers. So for the rest of this talk, if I mention a tree hopper, I'm talking about Vendusiae. My friend Farah, that helped me in the field for basically a whole summer, thought I was calling them nemipterans instead of hemipterans. So she'd always yell out, "Nemmies! I found nemmies!" And I'd be like, "What are you talking about, nemmies?" And then she's like, "Yeah, the nemipterans." And I was like, "No, they're hemipterans." So, and then here we are expecting them to have a double-sided arrow that's positive, presenting their mutualistic interactions. And you can see basically what happens is the ants come up, caress them, or massage them with their antennae, and that encourages honeydew production. So in 2012 was my first field season. I went out to Welder not knowing what a tree hopper looked like or what ants they were, because everything that I could read basically just like offhandedly mentioned, oh, on mesquite there's an ant that tends some homopteran, and that was it. And then, you know, I looked up homopterans, and I thought I was going out there to study fire ants and scale insects, because Ken did his work out there, and that's what he studied, and that's what I was expecting. However, no scale insects, but tree hoppers. So for all of 2012, since this was a new system to me, I walked around for the entire summer doing natural surveys. So over the entire summer, I censused about 120 mesquite trees for the presence or absence of the mutualism. So I'd walk up to a tree, spend 5 minutes scanning the tree, so walking around it. And if I found more than 2 aggregations of the mutualism, then I classified it as present. If I didn't, it was absent. I then quantified flower and fruit production, so counting flowers and fruits produced on the trees. And these are pretty large trees, so what I did was I broke the tree up into quadrants based off of the cardinal directions, and just to keep it systematic for the entire study, I always counted flower and fruit productions between north and west, and then multiplied it by 4 to get a representation of the entire flower fruit production on the tree. And then for summer of 2012, I collected all the data I could. So among other things, I also collected leaves from each tree, and then came back to UVM and taught myself how to do condensed tannin extractions. And that was when, you know, the department was super helpful. Brian let me borrow his sonicator. Probably doesn't maybe I remember, but I had it for a long time. Bill gave me a bunch of pipettes. So I had a lot of help working on the condensed tannin portion. But it provided with really, really nice preliminary data. Sorry, Emma, I didn't mean to say really twice. So in 2012, in this natural survey, just looking at ant or the mutualism presence or absence. So here, absent is the mutualism was not found on the tree naturally. Here, the mutualism was found on the tree. And then on the y-axis, we have flower abundance. We can see just from the natural standpoint that trees hosting the mutualism are producing more flowers than trees not hosting the mutualism. Likewise, looking at fruit production, mesquite hosting the mutualism produce significantly more fruits than trees not hosting the mutualism. And then looking at the condensed tannins, it flips. So mesquite hosting the mutualisms produce significantly less amount of condensed tannins than mesquite trees that had the mutualism absent. And condensed tannins is a secondary plant defense compound, and it usually acts as a metabolic inhibitor, but it's also been shown to stunt the growth of hemipterans. So it's a good indicator of the quality of the tree from the mutualist standpoint at the very least, if not for other arthropods. So we have this, I came back from this field season, cut up and super excited. I was like, I'm done. However, that's not the case because, as we know, correlation is not causation. So this is just what we're seeing. We don't know what the actual mechanism driving these changes or these differences are. So the potential drivers of these patterns could be just host plant quality. So these are really high quality trees, so they have the resources to invest in flower and fruit production. They can tolerate herbivory, it doesn't really matter to them. Or it could be the presence of the ant hemiptera mutualism. So the ants are indirectly protecting these trees through removing harmful herbivores. So I packed up my car and drove back to Welder the next summer to set up my experimental setup. And here for 2013 to 2016, I maintained what we call a press experiment. So I continuously removed ants from trees, not allowing them to recolonize. So it wasn't a one-time removal, it was a continuous removal of ants. So I haphazardly selected 100 mesquite trees that hosted the mutualism, because presumably if the mutualism is on there, they're all the same quality tree. So selected 100 trees, randomly picked 50 of them to act as an ant removal treatment, and then ant present was my control because that's what they were on, so unmanipulated control. And the way I removed the ants was I created these ant bait stations. So this is just PVC pipe that I drilled 2mm holes in them, so just large enough for the ants to go in and out. So this way it would mitigate the effects of the ant poisons on other insects, so it kind of encapsulated. And ants are very territorial, so once they find a resource, they'll protect it. And also these poisons weren't contact, they were the ant ingested, go back to the colony, and then feed it to the colony. So it was a really good way of removing ants. And I continuously recharged each bait station during the summer. And then when I went home for Christmas, I would stop by for a couple of days to recharge them. And this worked really nicely. So here we can see on the top graphs here are Camponotus. So these are the mutualistic ant. Down here is Vandusiae segmentata, the mutualistic tree hopper. So in 2013, present removed. So this is prior to ant removal. We can see that the trees had about the same amount of ants, had about the same amount of tree hoppers. And then a year later, through continuously removing them, we can see the ant removed trees had very few ants, and present trees maintained their colonies. And then in 2014, we also established ant absent trees. So these were trees that were naturally did not host the mutualism as a form of negative control. Because if we're thinking about equality and the preliminary data, the trees that didn't host the mutualism produced a lot less flowers and fruits than the trees that did. So this would give us kind of a comparison of, you're just a bad tree. You just can't produce flowers and nothing wants to eat you, so nothing's on you. And then we can also see that through the entire experiment, our removed and our ant-absent trees pretty much maintained their status quo. And then, sure enough, and this is something I wasn't expecting because there's very little life history on these on Vandusiae, is it turns out that they are pretty much obligate to having ants, to having Camponotus present. So once I remove Camponotus, the entire tree hopper community basically disappeared as well. So this strong connection between the tree hopper and their ant mutualist. So my first objective, what are the direct effects of the mutualism on communities? And then going into my second objective of the indirect effects of the mutualism on the communities. So focusing on the first objective, we measured the communities, or we measured invertebrate community composition by vacuum sampling the trees. So I modified a leaf blower / mulcher to act as an aspirator to suck up as many of the insects as I could. Previous studies that looked at ant hemiptera mutualisms on arthropods generally will, tanglefoot one branch and then collect the insects off of that one branch and look at basically what effect they have on particular herbivores or arthropods. But it's only really a branch by branch comparison. What I wanted was I wanted to see how were they impacting the entire community. So when I vacuum sampled the trees, I just didn't focus on branches that had the mutualism. I sampled the entire community, and I spent 6 minutes per tree vacuuming and got a really nice collection of insects. And this even kind of blew my mind of just how diverse these trees were. So over 3 years of sampling, I sampled about 37,000 individuals. I mean, there were a lot more, but those were left out of the analyses. And then these 37,000 individuals were comprised of 82 different families. So not even species level, these are just families. So I went to my lowest taxonomic level was families just because of time and identification. So back to just kind of recap, we hypothesized that the mutualism will have a negative effect on the arthropod community and at the family level and on functional guilds. So looking at family composition, what we find is this is 2013. So for all the figures, if you are kind of get disoriented, I'll have up on the top left, your top left, the year, and then whether or not it was prior or post ant mutualism removal. So 2013, prior to the removal of the mutualism, I sampled the arthropod community just to make sure that they didn't start off differently between my randomly selected trees. And this is a non-metric dimensional scale plot. So it basically plots your families on a plane, and you can just visually see how the families cluster out. And if what we're looking at here is as these clusters are highly overlapping, what it's showing us is the different treatments were pretty much had the same family composition. And then moving on to 2014, what we see here is we see a little bit more of our communities starting to separate out. So the green dots and elliptical are the ant present trees or the mutualistic present trees. The blue dots are mutualism removed, and then the red are the mutualism naturally absent. And one thing to note is for all of my analyses, I removed all of the Camponotus and Vandusiae abundances because if I left them in here, all of these would, the ant present trees would separate out just because the ants and the Vandusiae and Camponotus are basically highly abundant. So as I mentioned, and this is also another thing that was surprising, is on some of these trees you might have 60 different families, let's say, or 30 different families, and about 30% of the total abundance comes from just the ants and the tree hoppers. So I pulled those out as those were my treatments and didn't want them to impact any of the analyses. And then in 2015, everything kind of clustered back together on top of each other. So the family composition here wasn't very different. And when we look at the species richness, and then I use Chow-1 index as well as my diversity index, as this incorporates rare species and also estimates potentially species that you've missed. What I find is looking year after year, treatment between treatment, is the family composition is identical. There's no difference in richness. There's generally always the same number of different types of families. So that kind of goes back to the talk earlier Monday by Dan, talking about you have all these indices and it can all just be based on scale. So it could be a spatial scale, or in my case, maybe a taxonomic scale. That's why I brought up that question to him, was maybe, just the family level is too broad of a scale. Maybe if I went down to the species level, I might see differences. So then moving on from the family composition, going on to the functional guild composition, I basically broke down all those families, or binned all those families into 4 ecologically relevant guilds. And those guilds are non-tending ants, so these are ant species found on the trees, herbivores, natural enemies, so parasitoids and predators like spiders, and pollinators. And when we look at, and this is just a comparison of abundances of these guilds to each other. So the purple in this figure is Antihymetrin mutualism present, green is Antihymetrin mutualism removed. In 2013, prior to ant removal, we see that there's really no differences in the abundances between the guilds. When we move on to 2014, so 1 year after continuous removal of the mutualism, we start to see differences occurring in the guild composition. So here in the enemy guild, we see the ant apps. So these are in here the green are the trees that are naturally absent of the mutualism, had significantly less natural enemies than are manipulated. So the purple is ant removed, or mutualism removed, and the peach is the mutualism present. So we see that there's a significant effect of the treatment for the naturally absent, looking at natural enemies. For herbivores, we see that trees in which the ants were present have significantly reduced abundances of herbivores. And then pollinators, never really saw any differences with the pollinators, but that could have just been due to vacuum sampling is not the most efficient way to sample pollinators. It's a kind of a bias against them. And then we see here in the naturally ant-absent trees, there's a higher abundance of non-tending ants. And then in 2015, the only difference we see, or we found, was the enemy guilds were significantly higher in the ant-absent trees. And this is opposite of 2014, where they were the lowest amount. So still trying to kind of tease out why that happened. But in general, we see that there is a difference, or we see that the presence of the mutualism is impacting the guilds at some level. So in addition to looking at just the comparisons between each other. I also conducted correlations, so throw in all of the abundances of all of the organisms and conducting correlations on a per tree basis. And this is kind of where the story gets pretty interesting. So 2013, this again prior to ant removal, but again the treatments don't really matter for these because everything is pulled together. We see here these really cool kind of interactions. So the way this kind of heat map works is the darker the blue, the more positive the correlation. The darker the red, the more negative the correlation. And then if it's going to the right, positive, going to the left, negative. We see that Vendusiae and Camponotus interact with each other in a strong positive manner. However, they always negatively interact with the other guilds found on the trees. And this is, we found this in 2013. We go to 2015, we still see that interaction. And one thing to note is Vendusiae is generally always strongly more negatively affected or correlated to the guilds than Camponotus themselves. And that could be because they are kind of pressed on both sides of their abundances, where as ants increase, they're increasing, so they're going to have more of a negative effect. When ants are reduced and other things are eating them, knocking them off, so they're going to have more of a negative effect. But we see, again, in general, that the mutualism is just negatively associated with the arthropod community. And then in 2015, this was a really weird year. We had historical flooding, so my xeric scrubland savanna went to basically a swamp, where for most of the summer I was in shin-deep water. And I feel that definitely had a strong impact on what we were seeing. But even with this huge weather event, we still see kind of the same, the strength is different, but the direction of the interactions are still the same. And regardless of the weather, the ants and Vendusiae and Camponotus are happy together. So just recapping, from our first objective, we can't really accept the hypotheses that at the family level, the mutualisms are having an impact. However, we can see that they are altering the functional guilds to some extent. So the removal of the mutualism didn't have an effect on the community composition at the family level. So this could have been just not the right taxonomic scale that I looked at them. And also then the removal of the mutualism did alter the functional guild composition. So we found them to be highly negatively correlated. Herbivores were significantly less abundant when mutualisms were present. So then this could potentially impact the plant fitness. So not worrying about predators or non-tending ants, but looking at the indirect effects, the fact that these mutualisms did reduce the abundance of herbivores may potentially then have a negative, then potentially might have an indirect effect on their host plant quality. So that brings us to our second objective, where we were interested in how do these mutualisms impact reproductive success and quality. So if you remember from my 2012, we found that trees that hosted a mutualism had lower condensed tannins than those that didn't, presumably as a proxy for quality. And then when we look at flower count, so here we're going by year, 2013 to 2016, mean flower abundance. So here is prior to ant removal. Moving on, so 1 year after ant removal, we can start to see that the ant removed trees are producing more flowers than the ant removed. And then when we added the ant absent trees in the green, we see that they're producing significantly more flowers than the ant absent. 2015, everything is kind of increasing. So this is when we had the rains. So things were producing more flowers. Again, the ant-present trees were consistently producing high amounts of flowers, whereas the ant-removed trees produced high flowers, and then in 2016 crashed. The ant-absent trees kind of had this general increase in flower production. Looking at fruit abundances, again, we see prior to ant removal, the treatments did not differ. Moving on to 2014, ant-present trees are producing more fruits than ant-removed trees, and significantly more than the naturally absent trees. 2015, everything crashed, and I couldn't figure out what happened. And then I remembered having a conversation with another graduate student that was studying solitary bees around my field site. And so I emailed her. I was like, hey, remember in 2015 we had all that rain and you were mad because you had to come back for another field season because you didn't capture any bees. Do you mind sharing some of that data with me? So she sent me the figure that they were getting ready to publish. And sure enough, from her data sampling the bee community, she basically in 2015, and this is from Sarah Cusser, what she found was in 2015, the entire bee community, richness and abundance, was non-existent around our field site. So therefore, regardless of flower production, if there's no pollinators, you're not going to have fruits. And then from some papers that I read that looked at flooding events on bees, because these are ground nesting bees, so they presumably all drowned. It takes several generations for the population to rebound, and that was why she had to go back, was to sample some bees to do some genetic testing to see if those bees were left over from the previous rain or did new bees come in and colonize. And then looking at percent nitrogen, so I got funding from the Carolyn M. Glass Award to test nitrogen in the leaves in 2015. So I randomly selected a small subpopulation from each treatment to test percent nitrogen, and what I found was ant-present trees produced the most amount of nitrogen compared to the ant-removed and ant-absent trees. So again, ant-absent trees always, producing, always kind of in the low end of the quality. Ant-removed trees are in the middle, so we kind of see the same kind of reflection of the quality to the flower and fruit production based off of our treatments. And just to kind of visualize it, this is mean abundance of flower and fruit production in 2014. So naturally absent and the ant-removed trees, the differences we see there, we can potentially attribute to quality. So these are just higher quality trees, so they're producing more flowers and fruits. And then when we bring in the ant-present trees, the difference we see between ant-present and ant-removed is due to the mutualism. So regardless of, you can be a really healthy tree, but if things are continuously coming after you to feed on your flowers and fruits and so forth, it kind of caps off your amount of your productive success. So therefore, by hosting the mutualism, you're able to presumably maximize your production. And then the difference between the ant naturally absent and present could be this additive effect of the mutualism and quality. So, and we see that also with that line graph, that the ants that remain, those trees consistently produced more flowers. And one thing to note is we see here that these ant-removed trees, after 3 years of losing the mutualism, are doing significantly worse than their counterparts where they started off equally. So presumably with the loss of these mutualisms, the trees aren't able to maintain their high quality status or reproductive success. So herbivory is huge. This is a chrysomela beetle, so they do kind of a double whammy. They not only feed on the flowers of mesquite, they also oviposit eggs that then burrow into the seeds and feed on the seeds. So herbivory can have a really strong interaction or impact on mesquite. And then even if they're not feeding directly by chewing, we see here, this is a chorid that is piercing the stalk of potential flowers and basically sucking all of the phloem from them. So from our second objective, we can kind of support this idea that the mutualism is having these strong indirect effects on host plant quality through looking at condensed tannins, percent nitrogen, flower and fruit production. So we're just up against the clock, so just quick recap, mutualisms seem to not have a strong impact on the family composition, but do alter the community at a guild level, so function kind of ecologically relevant level. And from that, we see an indirect effect feeding back into their host plant through reduction of herbivores, and therefore why we see this increase in flower production and quality as well. So with that, I will take any questions.
Great talk, Nabil.
Thank you.
So as somebody who's used to seeing p-values that are much, much greater than 0.05, I was wondering, other than taxonomic scale, have you thought of any reason why basically this mutualism might just not have an effect on community composition?
So I think a lot of it, so most of the studies that have kind of done slightly similar things where they're not measuring the entire community, they do find strong effects of the mutualism, but I think the reason they do is because, like I said, they'll have one branch that has a mutualism and then another kind of corresponding branch on the same tree that they remove the mutualism, and then they'll sample the arthropod community here, and therefore this one has the mutualism, and therefore there's lower whatever organism they're looking at. Whereas for me, I sampled the entire tree, so instead of having these like kind of per branch comparisons, I'm collecting arthropods on the tree where the ants may not be there. Mm-hmm. So it'd be equivalent to their branch that they removed the ants from. So with my data set, even though I didn't find any differences at the family level per se, what I'm finding or what I'm seeing, I think, is more informative because it's looking at not just the direct impact of the mutualism on the branch they're on, but the indirect impact on the community on the rest of the tree. So even though those differences aren't completely different, there's still differences in the abundances. So, and again, this is where biology and statistics meet, right? Is, maybe they're not statistically significant, but from a plant reproductive standpoint, having 2, 3, 4 more flower herbivores or seed predators might be impactful from a reproduction standpoint. So I think for your question is just that maybe not taxonomic scale, but just the scale of the sampling.
Yeah.
So the interesting thing about it is that you're saying it protected from herbivores, but the actual mutualism has a bunch of herbivores that it's protecting. So I'm just curious if did you see if the plants themselves, you're calling these plant quality, sometimes to me, from like agricultural perspective, having lots of seeds might be a stress plant, right? So did you notice any increase in biomass? Were trees bigger that had fewer mutualisms on them because they were able to put energy rather into seeds to increase their evolutionary potential too?
So I tried to maintain all of my trees in the same size range to kind of mitigate size effect of the tree.
Or greenery each year?
Yeah. No, one thing that I did measure one year was I put little zip ties at the beginning of the summer and then measured growth for, didn't find anything. So I don't, and yeah, the stress response of just kind of masting is interesting, but I think for these mesquite trees, they are there year after year. So I don't think they're highly drought tolerant, so that's what allows them to be so kind of invasive in that region. That during the summertime, they are, the greenest, brightest things out there. And after my first field season, towards the end, as you mentioned, being more green, I could walk up to a tree and be, 20 feet, 30 feet away from it and be like, I'm going to find tree hoppers in that tree. just because the trees that had the mutualism versus the trees that didn't have the mutualism, those trees were, you could tell they'd been hammered or didn't have a lot of flowers. Their leaves were, like you're saying, less green per se. I mean, that's all subjective, but I don't think, I understand the kind of, I guess the concern was it just being like a stress response, but these trees are so large that I think you would need a lot of just these huge amounts of the tree hoppers to really have an impact. I mean, there's there's work on spiratina and things like that that show that with aphids, they'll feed on the spiratina, and then once they'll hit their threshold, because then it's just not a good quality anymore, then they'll switch from making clones to dispersal morphs, and then they'll move on to another patch. Whereas in my study, and the mutualism is highly dependent on plant quality, so honeydew quality is correlated to plant quality. So if the plant quality goes down, then the honeydew quality goes down, and then the ants are like, this is not worth it, and then they turn from being a mutualist to a predator, and they'll just say, I'm getting more from eating you and protecting you, per se. So the fact that I see year after year these mutualisms on the tree is persistent, I don't think it's like a stress response for the changes in the flowering.
Okay, one more question.
Okay, sneak it in.
All right,
All right.
two more, because Joe has to have the
Yeah,
last
I-
question.
Oh,
Sarah, then
No, I
Joe.
don't have a question.
Okay.
You don't have a question?
It's so clear and complete.
So you see on ant-present trees that the trees are doing better, right? There's more fruits, more flowers, higher nitrogen. And so it seems like the conclusion is that on the ants present, you have this mutualism, and so they're jacked up on sugar and they're taking care of their herbivores,
Yep.
right? So the tree's getting the benefit because it's getting these herbivores taken care of. So is there any consideration that it's more of a direct impact of having ants on the trees? In particular, I'm assuming they're nesting at the base of the tree. So you talked about how ant nest can have big impacts on the soil properties. So is that a consideration that it may be the ants, when you take out the ants, it's not just killing the mutualism in the herbivore source, but also taking out the ants at their nests and you no longer have potential soil contributions to the trees?
Yeah, so I think for that is when I said they nest at the base of the tree, they're not nesting in the soil, they're nesting in the fissures of the tree.
Oh, okay.
So that's one of the things I found at my field site, at least, was trees that were under 2 meters generally didn't host a mutualism. And I think a big part of that is because when they're that tall or this short, there's just not enough, they don't have bark or stuff like that for the colonies to form. But when they get bigger, then the nests are usually found in like kind of the base of the tree, not the ground, but the actual tree. But then to that same point, I didn't show the data here. If I have it, I thought I had it in here. But what I found from that preliminary work was, as I mentioned, I went and I scanned the trees for the mutualism presence or absence. I did find trees that hosted Camponotus, but didn't host the tree hoppers weren't there. And what I found was trees that only had Camponotus still produced lower flowers and fruits than trees that hosted the mutualism.
Okay, well, thank you, Nabil.
7 years in 1 hour.
And thank you all for coming.
Thank you. Gorgeous snacks on the way out.
Oh, no, it's fine.